Then you'll get a link.
And then I'll get a link. And so I'm assuming now it's recording.
Yes.
Okay, awesome. Wow, welcome everybody to the Mycology Club, first Mycology Club meeting of spring 2025. I'm glad you all were able to make it, and you all survived the illnesses that are going around, and you braved the roads and the ice and the snow, and many factors were preventing people from coming to this meeting today. But we have with us Dr. Tyler Barzee from the Biosystems Engineering Department. He's going to be giving us a presentation about what he does and studies here at the University of Kentucky, which is very, very, very exciting. As you saw from the title of this talk, it's Beyond Mushrooms. So we're going to be learning all about filamentous fungi. So strap in, and it's good you're all seated because you are going to be so excited. Just a little programming notes for what we have coming up in the rest of the semester. This is today with Dr. Tyler Barzee. This is our cool event. This is us. Coming up on February 15th, we're going to be having a foray at the Pinnacles in Berea. So y'all can attend that if you want to see. We're going to find, see what we find. We're not looking for anything particularly edible, but we're just going to, see what is growing. And that's on Saturday, February 15th. Time will probably be around 11. And I'll send an email about that with the particulars and all of that. So on February 28th, not February 29th, we're going to be having a guest lecture from Dr. Brandon Matheny from the University of Tennessee, Knoxville. He does a lot of work on a very particular family, which I am forgetting the name of, but he also does research on kind of ethnomycology in the Appalachian region, which I think is interesting. He's not going to be talking to us about that, but he will be talking to us about his other research. Britt Bunyard is going to be talking to us on Sunday, March 2nd. I think in this room. They're both going to be, we're like in this room and then next door and then back here, I think. And Britt Bunyard is going to be talking to us. He is from Wisconsin, and he is the editor of Fungi Magazine. And so he will be here talking to us about what it's like to be the editor of Fungi Magazine, and he's also heavily involved in the Wisconsin Mycological Society. And I want to extend a thanks to the Bluegrass Mycological Society for organizing all of that, and we're just hosting them here. And I think that is what we have so far. At the end of April, if you want to mark your calendars, there's the Kentucky Mountain Mushroom Festival, and that should be the 26th and 27th. So we'll be there one of those days. But okay, that's enough about what we have going on. You all are here for our presentation today from Dr. Tyler Barzee. And we have swapped views, but no fear.
I can fix it.
Because, do you want to, I was going to swap, let's see.
No, I'll just swap.
All right, I'll let you handle it.
Yep.
We have Dr. Tyler Barzee from the Biosystems Engineering Department. Got his undergrad at Clemson, got his master's and doctorate at University of California, Davis. Does a lot of really interesting research here, and will be talking to us about what he does. So I hope you all are excited, and he's going to introduce himself. So I'll get up to Dr. Tyler Barzee.
Thank you. Just gotta get this. Guess we're a small group. I can probably, my voice carries, but this might help me not strain. All right. You hear me okay? Yeah, okay. All right, so yeah, thank you for the introduction, Aaron. And also thank you for inviting me. I'm a big fan of this club. And so I joined the listserv, and ever since I'm like, why can't I see all this kind of excitement and all the student groups that happen on campus? So I'm very inspired by this. So great job, and I'm really happy to be here and talk to you. I have no idea how to make the toolbar at the bottom go away, so guess it's just staying. We'll go with it. Yeah, we'll just go with it. So yeah, so I know we're very interested in mushrooms, as am I, and I'm a huge mushroom eater myself, but I kind of wanted to take the time to talk about something that doesn't get talked about a lot, but it is stuff that I do in my research lab here at University of Kentucky, and I've been working on for a while. And there's some, I think, hopefully interesting stories and case studies that you can see kind of a different dimension of this amazing organism that you like to eat and look at, and all the things that fungi do for us. So I have a couple case studies, 3 case studies actually, I'll tell you stories about, but they're in future foods, what I call it here, and materials, and some funded projects from places like the USDA and others for sustainability and other types of things like that. So enough about that. So this is kind of my background. So Aaron gave me a little bit of introduction there. And so I grew up, I was born in New York, and actually most of my family lives up in New York, but I grew up mainly in South Carolina, and I went to Clemson University for my undergraduate studies, and I went from Clemson out to California. So in California is really where I got very interested in fungi, but I was doing a research project out there that was in a different area, but then was able to convince my advisor to, can I get a chapter of my dissertation in with fungi stuff? And I was able to do that, kind of mixing some undergrad research that I had done in Colorado. So there's a connection with Colorado and my interest in fungi as well. But anyway, I have 3 degrees. All of them are in biosystems engineering, which is a field that I'm very passionate about. But basically, it's the engineering of biology, and it has roots very deeply in agricultural things and agricultural engineering. And so, the way that I approach the work that I do with fungi is really through the lens of like agricultural things. And we are the only department at UK that is in both the College of Engineering and the College of Agriculture. So my boss is actually the dean of the College of Ag, but all of our students at the undergraduate level are all in the College of Engineering. So I have both of these worlds, and it's very interesting. So yes, but as it's come up, so the first time I met Aaron, actually, we had some shared interest, I think, in classical guitar as several other people in the Mycology Society. So I like to always talk about how I'm a real human person with other things other than the work too. So anyway, these are the things that I'm gonna go over. And so I think that everybody in the room kind of knows a little bit about fungi. And so I'm probably going to skim over some of the intro stuff about the growth and the life cycle, but there's certain parts of it that are pretty relevant to what I'm going to talk about for my work. And so I might just focus on those. And I have some pretty pictures of those. But really, the 3 case studies that I want to talk about are stuff that's happening in my lab that, I give all the credit to my students for doing the actual work for that. And then I have the great job of coming up and just talking to you about it and bragging on them. So, but they're pretty cool projects, I hope. So this slide is the background of basically things that drive a lot of the work that I do in my lab. And so while I love to eat mushrooms and all of that, I am constantly very interested in ways that fungi and other microorganisms can help us as a society with this looming increase of population that we're going to have moving into the 21st century. With a huge increase of population, we have a huge increase in food production that we have to do that. It's actually one of the things that fungi can do quite well for us, I think. And then on the end with energy, not as much directed to fungi in terms of energy production things, but fungi are some of the, they're nature's degraders, decomposers, and they do play a role in that green line that you see in renewable energy up there. So, but anyway, the overarching question of a lot of things that I do in my lab, which is a bioprocessing and fermentation lab principally, is around the question at the bottom of that screen. All right, so things about my approach to the work that I do in my lab about. I said the word bioprocess. We have bioprocess engineering. So a lot of the things you're going to see me talk about happen in bioreactors. So if you've ever been to a distillery, you've seen a bioreactor where they're doing fermentation with yeast, which is another type of fungi that I'm going to mostly be talking about filamentous fungi here. But Yeah, kind of looking at different ways that we can basically use bioreactors to increase efficiency of things is one of the things I'm interested in. And then if we can take more things and put them into a bioreactor, it gives us a place where we can really take those flows, harvest the good things from it, and then take the byproducts and cycle them back. And so this kind of circular concept is what you see on the side here, which is something that plays into a lot of the work that I do with fungi, but other organisms. But as the decomposers of Earth, clearly fungi have this kind of built in their just biology. So um, these are the beautiful fungi. So I stole this slide from, if you can't see it, but at the bottom of this is the credit of this slide, which is my mycology professor from Davis. And he was a very inspiring person to me, and I took this just directly from him with permission. But he put together this slide of all the beautiful things that you guys were talking about, from the macroscopic standpoint of, all of that you can visually see when you go out and foray and so forth. And then over here in the microscopic, I mean, some of the things that I see in my lab when I'm looking at them under the electron microscope is just kind of, I remember actually the first time that I looked at my filamentous fungi under the microscope and my mind was just blown. It was like, basically that you're looking at like a forest of mycelium, and it was, yeah, pivotal moment for me. So anyway, they're beautiful and wonderful. Fungi are more related to humans than they are to plants. So I hate it when people talk about like fungi as plants, and especially I do some work in alternative proteins and things, and they'll just say, oh yeah, we'll just make that plant-based burger with mushrooms. And I'm like, no, that's not what they are. So they're actually closer to you than they are to a plant, and evolutionarily. So anyway, some of the things, they have their own kingdom. Um, I'll talk about some of the other parts of this slide here, but for me, some of the things that are important about fungi for the problems that I'm trying to solve are relating to their structure. So making use of the different forms that fungi grow in is very interesting. And then their reproduction. If I don't understand how they reproduce, then it's very difficult for me to grow them effectively in my lab. And then also how they disperse, and that has some things too. So yeah, okay, fun fact, they're thought to have evolved 900 million years ago. And if that number is wrong, then this is from the professor at Davis, not me. Anyway, okay, so the morphology wise, it's really interesting. So in my lab, I have both of the top two that you see usually, and sometimes I also have mushrooms. But mushrooms really are just mycelium. So the top one for yeast is what you're going to see in the bourbon distilleries. And I am kind of the yeast guy for the new Jim Beam Institute that's at UK. So do a lot of fermentation work, and like half of my lab is just yeast now. But I'm very passionate about yeast too, but I mostly want to talk to you about mycelia, which is made up of hyphae, and you can see the building blocks of mycelium on the microscopic level, those forests that I was talking about. But why they grow this way in the environment as this what's called radial mycelium growth, where they kind of start and then move out as like a circle. And you've seen this when you're going out. But why they do that is really for exploration, and it allows them to invade new food sources. So find food and then take it over and break it down. That's what they're very good at. But also some fungi can actually switch forms between the yeast and the mycelial growth form, and that has important implications to things like the pathogenesis of them too, which is when they have dimorphic growth, if you want the word of that. But I'm not going to talk about the bad fungi in this one. But so here's the life cycle of the mushroom. And the mushroom that you see is just a very small portion of the life cycle of that fungi. And most of the time that fungi is underground, most likely, and you can't really see it. And so it's only this like event that happens that triggers it to make that fruiting body where you are visually seeing it. But, the mushrooms are all around us, and really that mushroom is produced by the fungi for this purpose. So, useful to keep that in mind, that these spores are getting produced on that fruiting body and then being taken out by water and wind. So that's the purpose. But most of their growth is stuff that you can't see necessarily. And then they can do both of sexual and asexual reproduction. Most fungi can do both, but not all. And so that's going to have some implications earlier where I, may have to work with different forms of starting materials for growing these organisms, depending on where we are in this figure on the left. So don't worry about the vocabulary there, though. All right, so then, we talked a little bit about lion's mane over there. So beautiful lion's mane is going to come up as one of the ones that I've worked with. But yeah, the basically categorization of fungi for ones that can decompose out there in the environment, and ones that degrade with enzymes or through non-enzymatic means. And so if you've ever heard white or brown rot fungi, that's what that means. So one of the things I'm going to talk to you about is the use of fungi in making new food products. And in fact, I will also talk about mushrooms. But it's useful to know that humans have been eating mushrooms for a really long time. This is from a published study that I did, a review paper, 16,700 BC is what the historians have direct evidence of human consumption of volatile mushrooms. And so then fast forward and you have bread production, beer production. So this 11,000 BC is a cereal-based beer using yeast and others in present-day Israel. And they actually have artifacts of this that you can go and see. It's really fascinating. But then fast forward to 7,000 BC, and you have the emergence of agriculture. And so there was a famous professor at UC Davis while I was there. His name is Charlie Bamforth, and he was the Anheuser-Busch Endowed Chair of Brewing at UC Davis. But the first line in his book that he published is that he believes that fermentation, of which fungi were the main, or one of the main at least, yeast-wise, one of the main players in early human fermentation. And actually, if humans didn't figure out how to do fermentation, they wouldn't have been able to maintain static civilizations the way that they have. Because what fermentation allowed them to do was to store food. And so once you could store food or store even water, if you've ever had kombucha. Kombucha is basically a tea made with symbiotic culture of bacteria and yeast, right? And so that is basically a preservation technique, if you think about it. And so anyway, fast forward, we have cheese production, koji. So koji is going to come up here a little bit. This is a production process. It's famous in Japan, or originating there. That is a certain type of filamentous fungi plus yeast. But the first part of that is using koji, is what the Japanese very affectionately refer to as these filamentous fungi that are the first parts of breaking down rice and making soy sauce. Anyway, so I'll get a little bit more on koji later, but so this whole timeline, if you look at scale, then we have the present day, which is in this little box, which I've blown up here. And from the Industrial Revolution in 1760, and then when yeast discoveries by Pasteur and so forth, 1865 is when the US begins industrial mushroom cultivation. Then all this stuff started to happen, where we're learning more about biology and DNA and so much stuff. Now we have, I'm going to show you a slide in a little bit of all the different mushroom products or fungi products that are on the market now, and seeing this explosion of interest of fermented foods of one way or another. Single cell form cultured meats, all sorts of weird stuff now. And something that you might also be interested in, since you're mushroom eaters, is in 1986, the launch of corn mycoprotein, which QUORN. It's a company from the UK. Seems like you haven't heard of it. You should check it out. Yeah, you can get it at Kroger. You know, they started out as Marlowe Food, and now I believe it's just Corn is the brand. But 1986, they were growing. They actually have the world's largest filamentous fungi airlift bioreactor is in the UK, and they grow massive amounts of mycoprotein that they put in basically alternative protein like chicken alternative patties and mince meat and so forth. But that's been around since 1986 and doing very well. So anyway, all right. So why am I going to be talking about fungi in foods? The first is because there's a diversity of species and products that you can get from these. You guys know that because you've seen them all over the woods. But also this versatility, as far as I'm concerned, as an engineer, I'm looking to look at the tools that I have available and how do I piece them in the right place. So the morphology and the life cycle are really basically tools that I have to play with. If I understand those things, then I can do different things with it. So they're also established. So we make biomass from fungi all the time. We also eat their fermentation products, and not just eat them. So this is another thing that not everybody knows is that citric acid. How do you think citric acid is made? When you think of citric acid, what do you think of? Citrus, right? Oranges, lemons. But you would be totally wrong if you said that citrus or citric acid came from citrus fruits on the scale that we use it. It's in Kool-Aid flavoring packets. It's in your laundry detergent. It's in it's in soda. It's all over the place. Candy, yeah. So that is a large industrial scale process, a fermentation process with Aspergillus niger, mostly, but some others, to make citric acid at a large scale, and that is how it's made and how we make it. So we actually have one up in Dayton, Ohio. I took my lab there and looked at. It's like their fermenters are huge. It looks like you're walking into a city of them. But yeah. So anyway, 95% of citric acid industrially is produced there. Anyway, so when you eat them, one very interesting thing about a lot of fungi is that they have complete amino acid profiles for humans. Not all of them, but some of them do of all of the essential amino acids. And then they have chitin in their cell walls, so this is a dietary fiber for humans, for our gut. So then texture and flavor. So one of the things that's very fascinating to me with mycelium especially is being able to mimic muscle fibers. So the way that they grow is actually very similar to how animal muscles grow. And so that's why some of the alternative proteins made from fungi are so successful. And they also are clearly delicious. Yeah, going back to my first slide, being able to increase resource use efficiency is one of the nerdy things that I'm very interested in being able to use fungi to do. And then, of course, for alternative proteins and things, animal versus myco products question there. So here's some of the proof. You can look at these tables. I won't dwell on this. I know it's a lot of data, but you know, just some of the profiles of amino acids from different edible fungi, and most of these are, so Aspergillus niger, this is the one that I told you is the one that produces the citric acid. You would see this and you would say that looks like black mold because that's exactly what it is. And Aspergillus is also, so Ariza, which is right here, that doesn't make black spores like Niger, but it is the principal workhorse of koji used for soy sauce. But this also looks like a whitish or yellowish mold is what you would think. But it's still, again, that's how most of your mushrooms are growing most of the time anyway. Over here, this is a Fusarium. This is the one that's in corn, another soil one. They isolated this from the soil. And, but then over here, Rhizopus. Have you ever had tempeh? Fermented soybean? I think you'd like it. You should check it out. But Rhizopus Oligosporus is the one that makes that. It's like a fermented soybean from, I forgot which culture. Does anybody know? Tempeh. Tempeh. Yeah. Yeah, it's good. It's cool. You should check it out. You can sometime, it might be in like the sort of, I don't know, global aisle or something of the, grocery store, but yeah. So I found it out there. Okay, so then to their morphology. So this is where some surprising results, I think, for you will come up. So when you grow fungi in what I call submerged fermentation, it means that I have liquid in a flask and I am shaking it around, usually, okay? They make fungal pellets. Fungal pellets look like, have you ever had boba tea? So they look like boba. So this is, I don't know if you can see, but there's these little dots there. So they're like these little, yeah, like boba. This is how they grow, which is, I think, quite surprising based on the imagery that I've given you so far of mushrooms and mold. This is what they do when they're growing there. So they make these little hyphal pellets, and the mycelium is growing radially the same way that it does on a petri dish. But it has some implications to how they grow. It's like a different environment on the outside than it is on the inside of that pellet. And so you can see the breakdown here where if you cut the pellet open, it'll be like hollow on the inside because it can't get oxygen in there. So it just grows from the outside. So it's pretty cool. But anyway, pellets of filamentous fungi for me means that it's easy to harvest. Though for yeast, when my students are doing yeast work, these are microscopic single cells. Like they're 5 to 10 micrometers big, which is, I don't know, less than your hair. And you can't see them. So getting those and harvesting them is something that has plagued fermentation scientists for years and years and years. But I don't have to worry about that. These things are this big, and I just put it through some cheesecloth and I can harvest it. So I like that. But yeah, it has some other benefits for that. And then the media that I grow them in. So when I say media, I mean food for the fungi. And clearly, food is important for you. It's important for fungi too. So here's some things that are really interesting that you can feed to fungi. So just some examples of different things. As I've said many times, they are the digestive system. They can break down a lot of things. So here you have byproducts from pea processing, pea as in the vegetable. Baker's yeast, and then starch processing. Down here are almond hulls, which are similar to peaches, actually. They taste like peaches. But you probably don't know that the almond that you get is sort of like that you eat. Do you like almonds? Yeah, so you actually are eating like the inside of like the pit of the peach. If you open up a peach pit, you will see this tiny little thing. And basically, peaches are so related to almonds, but the almonds were just selected to have this really large kernel that you eat. And at the expense of the fleshy part, which is the usual part that we eat of the peach. So almonds have this like super sugary, very thin layer, which I call the almond hull. And then inside they have a very small kernel. But they they have a very large kernel that we like to eat. But actually these hulls this is like, I think 2 times as much hulls are produced as those little kernels. And so you have these giant piles of almond hulls sitting around in California waiting for you to do something with it. And so what we did is we're like, well, we can make a tea out of it. And that's what sugar extraction is. Just steep it in hot water and make a tea and then try to grow fungi in it. And it works really well. And there are companies now in California, one of them that I'll show you in a bit that I work with, that are doing this on a commercial scale now, trying to take those almond hulls and make them into products like this. But that's that circularity process. Look at that. So, all right, here's fungi in future foods. And just a couple nice graphics that I put in one of my papers. And so I wanted to brag about them here. But I'm going to be talking about some things that are maybe like you could think of high and low tech applications to different types of products that you can make from food or from fungi. But they're going to go across a variety of different areas. And this is just focused on foods for now. And so I have two slides that look very similar to each other right now. This is a fungi biotechnology company map that this group puts together maps every year. And so this is the map from 2023. And you see all of these different ways that we're using fungi in consumer goods. Jerky, have you ever had mushroom jerky? It's delicious too. You gotta have mushroom jerky. And then coffee, or like that four sigmatic or mud water and stuff from that has mushrooms in it. We have mushroom grow kits. We have ingredients like mycoprotein and things. Actually, if you look at the projections for fungal products, most of it is actually down here projected in health and beauty products because of all of the extracts from fungi, like kojic acid is this thing. It's supposed to help your skin and all of that. And so these, you see a lot of things there. For fashion, I'm going to talk about what that means. How could fungi, interact with fashion? Doesn't it make any sense? And then packaging, so making basically those last two categories about making materials. So that's from 2023. That's like less than 2 years ago, based on where we are. And this is 2025. So already there are 4 more categories added at the bottom, and these have been expanded with many more companies. The point that I'm making, and I wish that I could get this. How do I hide this? Don't know how.
Maybe taskbar settings. No, Windows 10.
Oh, it disappeared for a second. No. I don't know. Well, you can't see the bottom, but I can send you the slide if you're interested later. Yeah, there's environmental remediation, pet food. I just had one of my students graduate with her master's degree from my lab and then went to a pet food company, and her entire thesis project was on mycelium for food production, and house and home, and then agricultural things. So yeah, I think it's looking good for fungi moving forward. Yeah. Oh, wow, really? Goodness. Yeah, I mean, so lion's mane that we were talking about earlier. So my PhD advisor, she's getting a little older now, but she fell off of, like, she was in a parking lot and fell off a curb, hit her head and got a concussion. And yeah, her physician prescribed her lion's mane to help. So it was enough evidence for that physician to say this would help you in your recovery for that. But I don't make any claims. I'm not that kind of scientist, so take my with a grain of salt, do some research on that. But anyway, okay, so I want to talk to you about 3 case studies I promised you. So this work that you're going to see is done from Shuvaro and Lauren. Hello, you can come down here. Yeah, nice to see you. So we've got these two that worked on this project. Lauren is the one that just got the job at the pet food company, and then Shuvaro is still with me getting his PhD. But this is where we talk about some high-tech applications applied to mycelium. So I'm going to talk about cell-cultivated foods, which may or may not be something that you've heard about before. But essentially, what that means is that you're making food with cells grown in bioreactors. I've already proven to you that we can do that with fungi. So the question is, what can you do with fungi in that area? And one of the things that you can do is do 3D printing. You've heard of 3D printing? So I have a 3D bioprinter in my lab that I'm
Oh my God, Rowan.
and putting them into a bioink and printing out these precise structures that have a lot of different applications in making different food products. You might be able to take this somewhere and have kind of on-demand food customization where you can kind of dial in the food that you want and then it just prints it out for you. So the military is interested in this, NASA is interested in these types of things. people are interested in these types of things, I find. So I found myself learning about bioprinting of different types of cells, including fungi. And so the first one application of this I'm going to talk to you about is fungi and microalgae, so a type of food. So this one is going to involve this particular microalgae here. So that's just a flask that I have in lab growing cells. They look so why am I talking about microalgae? This is, I promised you, fungi talk. So does anybody know about the relationship of fungi and microalgae? Lichen, yeah, like it's super fascinating. Lichen are wild. But like fungi and microalgae, the point that I'm making is that they share like a history with each other. They know each other and share some niches in nature together, like through lichen, which basically are these indestructible forms of life that can grow on the back of rocks and get super dehydrated and still live. But it's this symbiotic relationship, a synergistic relationship between, or mutualistic in the biology terms of making CO2 from the fungi, and then the algae eats that and then makes oxygen that the fungi eats, in this back and forth relationship. So the reason why I'm going to talk to you about microalgae is because this magical thing happens in the image that you see on the left, where imagine I have a flask filled with just green, murky algae water, pond water, you know? If I drop these little boba pellets of fungi in there and shake it around for a little bit, magically all of the algae gets attracted to the mycelium, and this is an electron microscope image of those mycelium just loaded with little microalgae cells on there. And so around like 2012, people figured out that this happened when you put these two things together. At least the engineers figured out that this happened. And it was this like huge rush of excitement. Oh my God, we have had the hardest time harvesting algae from water for the longest time. And now I can just put these fungi in there and they just soak it all up somehow. And nothing else that we've tried does it this well. And still now in 2025, I would say that why this happens is still not completely clear, at least to the engineers. Still, still like when I do this, I publish many papers on it now, it just seems like magic every time you do it, but it works every time. And it goes back to some relationship that they have with each other, and the fungi just wants to be there. And so for me, it means that they can easily harvest them. But also, these little red things that you see here are pigments from the microalgae, they may called astaxanthin, which is an antioxidant. And that algae is called Haematococcus, which in its Latin term means blood berries. So they turn blood red. So now when I'm talking to somebody that wants to make an alternative protein product, it's useful for them to be able to make a product that they can dye red. And now I have a great method to dye it red and give all this nutritional benefit from basically eating a microscopic plant with your fungi. So it's a pretty healthy food, actually. But yeah, so this work that you see in the center was funded by the USDA for like a future kind of food involving this bioprinter that you see on the right-hand side. And this was to make basically a type of you could think of as like similar to a salmon fillet, which is what you see here, which fun fact, salmon is the color that it is because they feed it astaxanthin. So it's from the microalgae that they eat. So I've cut out the middleman by doing this. But anyway, so that type of concept is getting used in industry. Here are this is a company that is licensing technology from me to make cultured caviar. So they're making caviar with fungi, these little boba with sturgeon cells in it that taste delicious. And I have been to tastings with Michelin-starred chefs that are eating this stuff and love it. And then also some of my colleagues in Spain are immobilizing yeast in the, are basically catching yeast into the filamentous fungi and using that for different types of things or applications in wine, beer, and production, all sorts of amazing things. So it all just kind of goes to the point of, if you can imagine it, like fungi can probably do it with this. I never thought that I would show like a slide with an actual product from a company just making these little boba into something cool like this that people want to eat. Anyway, okay, so then to Lauren's project, this figure is from one that I proposed to the Department of Defense for being able to extract proteins and different things from protein and the cell wall material from the fungi, and basically make hydrogels that you can make other structured products with, and that you could do all of that in one place. Like if you needed it in the desert or in some remote location, you'd be able to print out these products. So Lauren did this, and basically these are protein extracts that you see from, that's Aspergillus awamori, and this is Pleurotus, that's your oyster mushroom down here, so both of those. And so we made flowers and protein extracts from it. You can see some weird things happen too, where these like turn black in some cases. I have no idea why. I haven't figured out the chemistry of all this yet, but basically the proof of concept to be that you can take these proteins and make a thing that's more or less similar to like Jell-O, and then print it out into different types of products. And that you can do that from, in the case of soy sauce, the organism that makes soy sauce, and the organism that makes your oyster mushrooms. And then I'm really bummed that I didn't include the bottom two of this, which was wood ear, which you talked about at the beginning. So wood ear is not protein because they have very little protein, but all that stuff that makes them so jelly is these polysaccharides that are in there. But anyway, we included them as well. Anyway, all right. So now case study 2, bourbon shrooms. Talked about the Beam Institute. This is, I don't know if you're UK fans. Hopefully the people that are affiliated with UK are UK fans, but this is a super cool thing. This is affiliated with the College of Ag. My lab is in this building, just like 200 feet away from the front door of that. But that's the world's largest teaching distillery on a university campus. So that's pretty cool. And so they began distilling last year, and the first experiment that went through that was stuff from my lab. So like my students, now their barrels are aging in the warehouse, and it's like a mind-blowing thing for me as a faculty member, a young faculty too. But yeah, just really inspiring to me. But you probably know that bourbon is a big business in Kentucky. And if you need some facts, that there's more than 2 barrels of bourbon per person living in Kentucky, and that it's increased a lot. But the real thing that is the circularity argument that I'm going to come back to for fungi and how they play a role outside of the yeast in making bourbon is this. So that top right product that you see is made in quantities 10 times. For each gallon of bourbon you make 10 gallons of that, which is essentially like porridge, oatmeal sort of consistency, 1 billion gallons of that. And with the expansion of the distilling industry, the bourbon industry in Kentucky, looking to increase that quite a bit. It's a difficult material for the distilleries to work with. And it's also a difficult material for farmers to work with. They take it and feed it to cattle, and that does work to some extent. And the problem is that we currently have the lowest cattle herd in Kentucky since 1960 right now. And so basically, as the bourbon industry is going up, the cattle has been contracting. So what do you do to get rid of this material? You can't just put this on the ground. The question was, what do we do? First in lab, we were like, okay, we're gonna go out and get a bunch of samples of stillage from these different distilleries. Some examples of that are what you can see at the top of the screen, but the question really is about how do the contents and things that are in these stillage, clearly these are different, and it depends on if they're making bourbon or they're making rye whiskey or they're making wheated bourbon even. This has a lot of characteristics to it. fingerprinting of the stillage. I'm not going to go into that. But so back to this, all the different ways that you can use fungi in this. Basically, we went through this list and said, what do we want to focus on to upgrade stillage into something? And a lot of different ideas came out of that, including the ones that you see on the screen. But the one that I felt like I needed to just figure out first was the thing at the very top, which is bourbon shrooms. So we got funded by the UK Office of Sustainability to upgrade stillage into what we call bourbon shrooms, which is essentially, this is oyster mushroom. That's what it looks like. Okay, that's on a plate, just growing. You would say that looks like a mold, right? And this is lion's mane, and this is shiitake, right? But then we eventually get the beautiful mushrooms. Those are clearly the oyster mushrooms at the bottom. But this was basically a exercise of trying to figure out how to optimally grow these things and how to make them happy. And so then we made a grow kit of bourbon shrooms, which was a fun project. And we imagined that they would have this instruction book. I don't know if you guys have like done the mushroom grow kits before, but they're a lot of fun. And we were exploring this because basically it's a clever way to get people who are visiting your distillery to take away your stillage, which is your trash. Just say, hey, here's my trash. Pay me $25 for you to take that away. And is an interesting thought, but consumers also get a cool thing from this, which is the experience of seeing what it takes to grow mushrooms at home. So I have some of these at home, and just stick them on top of my fridge, and you spray them with water once a day, and they grow these amazing mushrooms out the front. So I was really kicking myself. I forgot to bring my mushroom bourbon shrooms grow kit so you could see it, but it's really just packaging, because inside of there is just a bag of colonized stillage, which is the same as any mushroom grow kit. But we did a lot of branding, clearly, of the outside. My lab manager, Zach, actually has my signature there, and that says Master Mushroomer instead of Master Distiller. But the rest of it is somewhat, reminiscent of a bourbon bottle kind of thing. And we haven't quite been selling these yet, so a lot of people have asked us if we're selling them or not, but I just don't have the bandwidth as a professor to run a company like that. But anyway, yeah, if you're interested in doing the legwork for me on that, let's partner up. But anyway, so this was a fun project, and it was an educational thing to also teach people about growing mushrooms. So common question that I get on bourbon shrooms is, do they taste like bourbon? And the answer is right. No, they don't, because bourbon doesn't taste like bourbon until it goes into a barrel, right? But we had an interesting question, could they taste like bourbon? Actually, sometimes when I'm cooking mushrooms, I actually do that thing at flambe or something where you put some bourbon in at the end and it catches fire. And bourbon flavors and mushrooms actually go together pretty well with some dishes. So we submitted, we got rejected the first time, but I have currently another proposal into the USDA to help us figure out how to get some flavors from a certain mash bill at the distillery that says makes this flavor of bourbon. Then if you can get those into the mushrooms that they grow in their kits, that would be pretty cool. And then I could teach people about stillage and the connection to this product that they know through the beverage, but do not have any connection to the waste product that it generates and how that could be used to make something cool like mushrooms. So anyway, we're working on that. I'll let you know when the results come in. But yeah, working with some other people on the campus to look at the breeding or the sensory, basically getting people to taste it and say, does it taste good? Yeah, anyway, the last case study that I want to talk to you about, running out of time and I don't want to keep you, is pure mycelium materials. So this is the connection to those things that you probably weren't expecting at the bottom of the application slide. And this is Kea, fantastic student in my lab. So pure mycelium materials are a type of material. I would classify them there as being two types of mycelium materials. The first is a composite one, which there's an example of a composite mycelium material being used as a that is a package for something. I think some kind of skincare product, or something. But imagine that. So that is basically got the texture of like a fiber board or a styrofoam kind of thing. And that what you're seeing right there is basically a grow block for producing mushrooms that they've cut into a slice and pressed it down. That's all it is. It's all those little particles of sawdust have been glued together with mycelium that's growing through it, making that white structure that you see. And then you just do a little bit of processing and mold it into the form that you see there. So yeah, in a landfill, I mean, it could still be, most of them are not still living, so they do like a heat treatment thing, so it's not still living and breaking down stuff itself, but it's composed of biomass that is biodegradable. So yeah, in a landfill would do that. You could also compost those types of things, that would be better. But okay, so down here are pure mycelium materials, which are basically composed of completely fungi and do not have the what I call substrate particles, but it's like whatever you use to grow the fungi in there. These are some examples of things you can get from that. Mycoflex here, this is that's a material. This is a like, actually, when you put that in a frying pan, it looks like bacon. Cooks down to like bacon. And then over here, which is most of the excitement for this type of material, which is in leather type materials. So these materials are, they're now being produced commercially in the United States. There's a company called MycoWorks, that was a California-based company that actually just built a huge processing plant and manufacturing facility in Union City, South Carolina, just down the road from my hometown, actually. And I was, even though I have the connections to South Carolina and fungi and all of that, they still won't return my email. So I've never visited it, but it's there. And then most of the things that you're seeing here are from either MycoWorks or Ecovative Designs or Bolt Threads. And they're mostly making these materials that are very similar to leather type of applications. So MycoWorks, for instance, just announced a huge partnership with Cadillac. They're making the leather interiors for Cadillac cars with pure mycelium materials that they produce in South Carolina. And one of the selling points, interestingly, is that like, you know, there's all the ethical things around like, should you grow an animal to slaughter it and then eat it, all of that, and you know, vegetarian and vegan implications of that. But otherwise, like there are just things that you can not do with a hide of an animal that you can with mycelium. And that was one of their main selling points, which was pretty cool, was that with the animal, you get a hide in a certain shape, right? And if you need a chair for your car, your upholstery for your car, that's not in that shape, then you're going to be cutting it out, right? And piecing things together. But in this case, you can actually grow the materials the way that they do in any shape that you want. It can be, they have images like of basically, you know, to the end of this room of just a sheet of mycelium that they've made in one continuous layer. And it's really wild. But anyway, you know, some other Lululemon and Adidas and Hermes, all these different things. So what it looks like when you're making these things. So I've never been able to visit these facilities. They're on lockdown, but I want to. What you're seeing here up in the top left is that an example of making that really long sheet. That's how they do it. And in picture B at the top is what they are ultimately growing. It's almost like foam while it's growing. And how they do that is, as I understand it, is they inject carbon dioxide into the substrate and it goes up and travels up, and that signals to the fungi to like escape, run away from the CO2 and go up to the oxygen that's up here. And so they just keep growing upwards into this aerial mycelium that you see there. But anyway, that's the type of thing that you're looking at. These are reminiscent over here of actually how industrial scale button mushroom production looks like. So I took a field trip out to Pennsylvania and looked at some of their large-scale mushroom farms, and they look very similar to that and how they grow them there. But yeah, different types of threads and things that you can make outside of leather with the pieces of the fungi, but this is, I'll move past that. And yeah, this is just showing you some things about environmental impacts and looking at mycotextiles to basically reduce some of the waste of making the clothes that we're wearing, because 60%, or I think 70% of fiber that's made for our clothing and stuff actually comes from petroleum sources. And so the question here is, if you can make them from fungi, would that be better for the environment? Anyway, so one study is out there that has tried to quantify that, and Rishi, this is MycoWorks. Yeah, so this is on MycoWorks Rishi product. They have trademarked Rishi somehow, but they have leather over here. This is for CO2 equivalents, so like climate change, looking at that impact versus their Rishi and finding much reduced for there. But I can't validate that, but I wanted to show it's the only study I could find of that. So for Kaya's actual project, she was asking some of these questions about basically making these types of materials. So we wanted to make them in our lab, and everything that I showed you on the past 2 slides is inaccessible. It's not in the scientific literature, it's in patents. And so I can't really figure out how to do any of it. The patents are, I don't know if you've ever read a patent, but it's not fun. And so basically I go to Kaya and say, we need to make these, how do we do it? And so we figured it out by answering the questions that you see here. So we used here, again, you're gonna see Pleurotus, is that oyster mushroom, and then Rhizopus, which is the tempeh mold. That's what tempeh looks like over there. So, and then we just processed our stillage through what you can see here, and then we grow the mycelium material, and we have this little membrane that we just take off of the reactor and just peel off the material. I'll show you that in a second. And then the last one, this is funny thing that scientists do is we say the aerosolized humectant. This is a fungi lotion. So looking at if we added these things, if it would help them stay hydrated and grow better. Anyway, all right, and then some fancy instrumentation that another professor helped us look at that actually measures distance, and so we can constantly measure the height of the aerial mycelium of our material that we're making, which is pretty awesome. That's what the output looks like, but that might be too nerdy in a different way. Okay, so the experiments that were performed is what you can see here, looking at various parts of the cultivation process. But again, I don't want to dwell on that, and I'd rather just show you the pictures of the results, which is here. So this is asking the question, if you pack the substrate, so the stillage, if you pack all those stillage particles together like really tightly, does the fungi have a harder time growing into that? Because what we want is for the fungi to not grow into the stillage. We want it to grow outwards up into the air to make that huge aerial mycelium. And so we just packed down the stillage, essentially increased its density greatly, and then grew it. And so this is what you see here is that it did start to, we saw some evidence of increasing the height of the mycelium material that we would get from that. But even better was basically giving the fungi something to grow on. We call it like external scaffolding or something here, external support. But essentially, like we found out that the fungi, when we're growing them in lab, is always growing on all of our glassware. It's like this tough thing to just like wrangle in. They're crazy, they grow everywhere and do all this. So all we did was just give it more access to this glass in the container, and then saw what it did. And this had a huge impact if you gave them a lot of area to grow. Basically, they grow up the glass, and then they like cross-link with each other, and then they use that to grow up more and then make more layers. So they made quite a lot of height there than what I showed you on the last slide when we did that. And so again, going back to the beginning of this, where like the engineers like can look at fungi and we don't know much about the genetics and these fancy things like that, but just looking at how they grow and how we can harness that for something like this, like, oh, they grow on glass. I bet they'd climb up that and do this thing that we want. And it was really exciting when we got that result. And then the other thing was we looked at the papers of research around tempeh, because tempeh, they don't want the spore of these black spores. That's a pretty ugly thing. You wouldn't want that. And the reason they ferment tempeh on soybeans, which is high protein. And so we thought, yeah, there was a paper that said, well, the high protein content is actually what suppresses the sporulation of the thing, which is why you don't see it in tempeh. And so we're like, okay, we'll try that out and do nothing to our stillage and keep it in the way, the form that is, that's carbon-nitrogen ratio. But essentially, this is, should we do a lot of work in changing our substrate as we would to grow like oyster mushrooms, or should we just keep it the way that it is? And keeping it the way that it is what worked really well here. And we were pretty interested in that result too. The more that we can suppress the spores, the more we can help them keep their exploration phase and not go into reproduction phase. Anyway, so, and then this one is about the fungi lotion, and I'll say like the results are mixed on this. It wasn't totally clear to us that it had like a big impact on their ability to retain moisture or grow, but it did seem to help them make more dense biomass in that mycelium, but that's sort of a work in progress. We're not really sure why this happened. But anyway, so the last thing that I want to talk to you about is future fungi projects that are in my lab. So I've talked about a lot of them here, but one of the most exciting ones is an undergraduate student in my lab. He's a senior and he's a triple major, and which I don't understand, but he's in biosystems engineering in my department, and he's also in agricultural and medical biotechnology. And then third is, guess what his third one? Any guesses? You will never guess it. It's violin performance. It's like, do you just want to stay in school forever or what? But yeah, he came in and so sake production. Have you ever had sake? So this is another way for you to appreciate fungi. Sake is a Japanese process. Koji here, I've used that word. Those are those organisms that go in there to break down rice. And then they break it down just like we do in bourbon production, and then we pitch it with yeast and make alcohol. But over here is what you see is there's a press. And so actually in Lexington, we have Void Sake, where not many cities have a sake brewery. So we're pretty cool that way. And the guy that runs it, his name is Justin, and he's fantastic too, but he came to us. He has a bunch of lees, basically leftovers, and said, can you make Kasutori's shochu? Shochu is a distilled spirit from basically a sake-like origin. And so Ben has been doing that, and that is a picture. I put it here because that was 2 days ago. Ben began in my lab with like a little lab scale, flask doing this and just saying, oh, what do I have to do to make this? And then he scaled it up. We've done 3 rounds of sensory on spirit that he's produced at the Beam Institute. And that's him 2 days ago pitching the first Kasutori Shochu ferment at Void Sake in Lexington. They're going to make a product, and they're going to actually take that on a trailer and take it to a distillery that's going to distill it for them and make a product. And I'm like, this is such a cool thing of my job. But anyway, so I wanted to brag on Ben there too. Anyway, so just wanted to acknowledge all my lab and the people who did all of this work. And then this is the picture that we took of my research lab a couple of years ago at Primient. That's the citric acid production plant in Dayton, Ohio, where we visited. So yeah, I don't know how much they give tours to the public, but that was wild and really cool stuff to see. So anyway, fungi is all around you, and it's super fascinating in ways that you might not even know. That's the long and short of it for that. So thank you for your attention, and sorry to go a little bit over here, but yeah, it's been fun talking to you guys. Thanks for having me. And yeah, do you have any questions? Yeah. Which one? Yeah, okay. They have actual logs still? Used to.
The original, it was close to the grandkids from an original distillery that shut down to open the property back up.
Mm-hmm. Okay.
So, but originally, like a great grandfather had a log still.
Yeah. Yep. Mm-hmm. Okay. Yeah. Now it's the same copper. Yeah.
It's made to look like a log.
Yeah.
But, so do you have any distilleries that are fighting on that, like doing that?
On the stillage, so yeah, so a lot of the distilleries are super interested in stillage. So, all right, since you know about the bourbon industry, so basically stillage for the big guys in Kentucky, so think Makers and Beam and Buffalo Trace, like they all have dry houses, right? Do you have a dry house? Right. Yeah.
Sell it. Now it's such an abundance of it, they have to pay to get rid of it.
Yep. So yeah, multiple trucks per day, probably $4,000 per truckload or something is what I've heard. So this is a multi-million-dollar problem for distilleries on a yearly basis. And so big guys have dry houses where they separate the stillage. They have a centrifuge, and then they have a dry house that basically makes it into dried distillers grains, which is similar to, well, it's a cattle feed, basically. Okay, so you can ship that around a lot more cheaply than you can something that's 95% water, right? So that option is not accessible to most of, and we have like, I think 60 or 70% of our throughput of bourbon production in Kentucky is from craft and medium scale distilleries. So like down there, I forgot where Log Still is, but probably got like Danville and Wilderness Trail up there. Oh yeah, okay. So you got ours, okay, got it. Yeah, so, okay, yep. Yeah, so, but anyway, there's a lot of production from, people who can't afford that. And so that's where most of our interest has been, is like, can we do this? But then, the problem inevitably is that you've got a solution, but like, can you scale it and all of that? And making bourbon shrooms, I did the calculation. If you were to just grow on all of the stillage that's available from one distillery, and the yields that we got for mushrooms, of oyster mushrooms, if you did that, you would produce the entire US current production of oyster mushrooms from one distillery's stillage. So clearly you're going to saturate that market very quickly. You have to figure out something else. Yeah.
Processes are you using for spreading?
Yeah. Well, so at least in the lab, and this is the same if you were to scale it up. That's right. So, yep, you got to take it. Where do I show? Oh, I forgot that slide, or didn't include it. But anyway, yeah. So in order to get to here, you've got to basically separate solids from the liquid, and then use the solids. We mix it with a little bit of, so we use sawdust from a cooperage, and they've got, tons of sawdust that they're giving away, but that's a great carbon source, and it increases the C to N ratio that I talked about a little bit. So that's a good thing for oyster mushrooms and also for lion's mane. And shiitake, but anyway, to various degrees. But so we mix that together, and then we do a little bit of buffering capacity into there with basically bicarbonate in there, baking soda. Put that in to just help the pH stabilize a little bit. Stillage is acidic as it comes from the distillery. So yeah, you got to do those things. And then, of course, keep it sterile, and then have a good culture that you can inoculate it with, and have means to maintain at least sterile conditions for that, in this case, grow bag, through the colonization of its life. Or else you get what you also saw, which was mold growing on the, around the mushrooms on the trees, happens in the lab. And it can be quite a huge issue in the lab, or in any controlled environment where you're growing massive amounts of them. Yeah. So for liquids, we actually use the liquids. So at the top of the screen, those are all mycelium pellets that we've been growing with the thin stillage. So when we separate the solids from the liquids, we get the pellets grown up over there. That's just grown in thin stillage. And so then that's our inoculum. And so that is just a convenient way for us to make use of the water. It's, still you're going to have leftovers there, but at least it's removed some of the carbon and things that would be bad to put it down the drain, or at least, yeah, help with that. But yeah, I definitely, so I took bourbon shrooms through a boot camp process at UK where they want me to make a business and be able to pitch it and stuff. And this is where I have like real conflicting things with my job as a professor and a purveyor of truth and knowledge, and then being a business person that just wants to, oh yeah, you've got your problem, I can solve that. Yep, I can do that, and I can make you tons of money by doing it. And it's like, I have to be real with people that like, I definitely do not have all of the answers for how this whole system would solve the problem. I think it could contribute to the solution, but the truth is that I think that there's a variety of different things that have to come together to make things work, and it's not going to be a one size fits all for these distilleries. What works for Log Still is not the same as everything.
Tankers a day, it's
Yeah.
a smaller operation, but
4 tankers per day is.
Separate that, turn it into a product ready to sell. I mean, you would get behind on.
Yeah.
You have to have a huge operation to even keep up.
Mm-hmm. Yeah.
You'd have to have a big facility to keep up with a small operation.
Mhm. Yeah, you would. So one thing is that you might be able to kind of centralize that facility. So that's one of the things that I saw when I visited Pennsylvania for their Pennsylvania is like the heart of mushroom cultivation in cultivated mushrooms in the U.S. And I don't know what the connection is, but the Amish and Pennsylvania Dutch kind of thing that just grew this. And so these are absolutely massive mushroom operations. But they have centralized facilities that make their compost and that make their, it's called casing, but it's essentially like peat moss that's taken mostly from Europe and then prepared, and that's what cases the tops of the compost once it's fully colonized, and then the mushrooms actually grow through that casing layer. But those facilities are these gigantic centralized facilities that then take that compost material out to all of the different mushroom houses that are actively growing mushrooms. But it's pretty uncommon for them to do both mushroom cultivation and preparation of the substrate at one place. So maybe you could get a big operation that would take these tanker trucks, do solid liquid separation there, and make a really good product for substrate, and then be connected directly to a water treatment plant that they could take the liquids down to that and then keep the solids to go to mushroom cultivation.
I didn't talk about things getting sterile in the last day.
Yeah.
And why is it that so much abundance grows out in the forest where there's no control over sterilization or anything?
Well, it might be a better question for somebody else in this room too than me, but my take on it would be that mushrooms are filling a niche for us in the environment. If they're there, it means that they are finding sufficient food source and things around them. And so I guess they're still susceptible to getting contaminated with other things. Like stuff is always, but that's sort of what nature is this like giant war of different organisms constantly happening out there. But you just kind of see sometimes that things, and we actually see this, the best corollary I can think of in like the lab is when I have a petri dish. I might have inoculated that with something that I want. If something else gets in there, it will, start in a place that it's easier for it to take over. So it'll start over here in the corner and then sort of grow and start attacking the thing that I was growing. And eventually they might come to an equilibrium where they're like, okay, I'll take over here and you take over there. And we're just gonna coexist together. And so I've seen that happen before, but it's certainly not always the case. But it's a struggle. So I guess you're just seeing who's winning out. Right.
They did. About what they couldn't use themselves or even sell to other people. I don't know. So they came in and dug a big couple acre pit on the farm and just dumped it.
Wow.
And they got from the smell and the rot of it all, they've kind of got a spotlight on them, and I don't think they're allowed to do it anymore. But
Yeah.
I was thinking, like, if the ground would take away your moisture, and could you not mix sawdust and your ingredients into this big pit that's open air, and what would happen?
Yeah. So in that pit, so that reminds me of my time in California. I have a lot of background in dairy manure lagoons. And so I've spent many days on the edge of a lagoon taking samples through the night and so forth. And that's what it reminds me of, which is the topic of another potential solution for stillage is to dig a big pit and make a covered lagoon anaerobic digester. So a digester that basically does the same thing that happens in the rumen of a cattle or sheep and makes methane. So you're seeing this. There's big players in Kentucky and Tennessee that are doing this. We have a published study on doing anaerobic digestion and basically that it works with stillage. It's a good option. It's bacteria and archaea, has nothing to do with fungi, unfortunately, but that produces renewable natural gas and it stabilizes the liquid such that they can be used as fertilizer in most times of year, except for like clearly right now when you can't apply it to land because it's frozen. But yeah, that is one thing that you could do with that pit. I'd have to get convinced on growing culinary mushrooms that way, because one of the things is that fungi are slow growers. Like bacteria, like my lab is filled with bacteria and other faster growing things. And I always have to worry about them getting in and contaminating my stuff because if I get bacterial contamination in like that flask up there, it grows within like half a day. I see it get cloudy and I know that my pellets are not going to have any chance. So I don't know. That's maybe I've just seen too much. I'm jaded.
Covers even on decaying stuff. And there's actually some that will grow as the log or even a little. Some of them grow as it still has bark on it.
Mm-hmm.
And some grow the next stage as it's losing bark. And
Yeah.
some stuff will grow all the way down to being almost rotted into the ground. So it's like they're taking different stages of the decay.
Yeah.
I don't know if it's really happening that way. We're just seeing the time frame different, but it seems that way. Like some stuff will grow on newer.
Interesting.
Boundaries.
I'll tell you what, if you can figure it out with that, like do it.
Whatever materials they're feeding off of, they're getting access to it at different times.
Yeah.
Breeding at the same time.
Yeah.
I mean, it would be a lot. You might not see them all, but it would be a ton.
Yeah.
Versus here,
Yeah. This is a fun story because I told you about the thing in Colorado that I did. So that was how I got into mushrooms. You know how that happened was I was an undergrad researcher. I did not come up with this idea, but the lab that I was doing, we were trying to was called bioprospecting. So prospect for fungi. And where did they get the fungi from? It was from rainforest soil. So they went down to Costa Rica and took these like tiny soil samples. And I mean, yeah, just like what you're saying, it could be like a little batch of soil this big. And that turned into, I have a great picture that I did not know to include in this thing, but it's an entire biosafety cabinet of what I had to do every day was I had 100 isolates of fungi from like this little thing of soil. And I had to, each one of those was one single organism, and there were 100 of them. And I had to maintain the petri dishes of these things. And all of them were totally different, but they were all in there, but you would have no idea unless you did that.
When you're talking about them capturing methane off.
Yeah.
So there's actually one of those facilities, I believe, coming into Lebanon.
Lebanon, yeah.
And so what they are saying is they're gonna capture the gases off of it, and then what they're gonna be left with is gonna be like animal feeds. We actually had a company propose on to want to build property. And they're talking about our company giving them lots of land to set this facility up, and we're just not gonna have that cost of somebody coming in and getting our.
Yeah.
facility.
Mm-hmm. For your still boiler, yeah.
But they're claiming that certain algae or whatever that they're introducing, that they can actually have potable water.
I've heard this too. Yeah, so digested organics maybe that it's coming in. So yeah, this is all fascinating stuff. So Bobby, the CEO of Digested Organics, similar to me, like has a background in dairy manure, and we can't get away from it no matter how much we try to go to fungi and other cool things. It always comes back. But yeah, so that's so the anaerobic digester is one thing. That's a biological, that's essentially a fermentation. And it makes methane that takes off, but then you're left with digestate, which is it's a black liquid of like 95% water, just like stillage is. But it doesn't smell as bad. It's more stabilized. It's sort of like composting in some ways, but totally the opposite of composting in other ways. But it activates a lot of the nutrients or mineralizes a lot of the nutrients that are there and makes a fertilizer product. What Bobby's company, Digested Organics, comes in on the back end of anaerobic digesters and has a filtration unit. There's actually one of them already on site at Wilderness Trail. So you can talk to Pat Heist about their experience with it. They don't have a digester. They were just trying to use it as a stillage solution. I don't think it worked for them for that. But that they can actually make potable water. One of my projects funded through the EPA is operating in California. A digested organics system is there and basically makes clear water at the end.
Benefit of us being shared space, you know, being on property would be, yes, through the process, they have the water is heated already to whatever temperature.
Yeah.
They're hoping to pipe that in and be preheated for the boiler or preheated for other things, so we don't have huge energy.
Yeah, water savings and energy savings. It's great. Yeah, that's cool. So it might be a scale issue is they might need a centralized thing. I did some calculations on it for smaller. Yeah. Yes, exactly.
Don't have to. We're gonna be so close that we're not gonna have to.
Yeah, you just don't have to worry about your stillage. So, yep, that's the business model. Interesting. Well, yeah. Oh yeah, I mean lab tours for sure. Yeah, you can come. I'm really happy to do that all the time. We also do E-Day. So we have so E-Day is Engineering Day at UK, and it's like the big open house of the College of Engineering. And we are the mushroom table. So we're always teaching people how to grow mushrooms, and everybody's going from like robotics over here, and then it's like, hey, we're talking about mushrooms. And they're like, yeah. We have that kind of public outreach stuff that we do with the lab. That's just one example, but do that as well. But in terms of like lectures and classes, I've never had that happen before other than grad students in the lab. But yeah, I have like lectures and things that are also posted on YouTube. So one of them is similar to this talk that I gave, but it's called, well, it was a fungi food thing similar to this. And some of the slides are actually the same, but that's on YouTube that you can watch and so forth too. But
Just slides and stuff you had today, when we have access to these.
I don't know. Yeah.
Let's put these on YouTube.
Yeah.
There's a video.
We have links on
What?
we have links like on the mycology site.
Yeah, I'll email that out and then I'll people post it.
There's a guy that he is the, I guess, chemist, in-house chemist. But he's just charmed. He talks about a lot of stuff.
Yeah.
He knows growing mushrooms too at home, and he just full of knowledge.
Yeah.
He
Okay.
interested in knowing
You'd
about
like
it.
it. Awesome.
I know a lot. I know or two tell people about today, but just people not.
Yeah. It's great. No, it's great to see you guys. I'll speak to however many like want to see, but yeah, like I really appreciate you guys coming out, and I don't know if there's anything. Yeah.
About the, I'm assuming that you're doing about health.
Yes. So yeah. So with Aspergillus, so it's important to know Aspergillus niger, while it produces all of those, citric acid and all those things, products for us. Aspergillus niger also produces aspergillosis, which is a lung disease that is it's not common by any means, but if it's going to happen to somebody, it's usually to farm workers, because Aspergillus in the environment lives in the soil, right? Talk about that. It's in the soil, and if you're walking behind a tractor as it's tilling or otherwise being exposed to large amounts of dust from basically this tilled up soil happening, then if you breathe in a huge amount of spores and you're immunocompromised, it can make quite big health impacts to you. And that is also the same thing for pleurotus. So for oyster mushrooms, you might not know this, but yeah, oyster mushrooms grow a lot of nodding heads, so you do know this. But yeah, when you are growing those at scale, industrial scale facilities that make oyster mushrooms, they are wearing masks because oyster mushrooms make a ton of spores. I was that an oyster mushroom in my example there? The one with I can't remember what that was, but oh yeah, it was not a. Who can identify that? That's the question. Yeah. Okay. Yeah. Emily. Yeah. But anyway, yeah. But okay. So there's an equivalent version of that for oyster mushroom sporing. It's quite a lot, and yeah, you gotta check.
Hydrated water when you get them out of their environment. They are usually very, very wet to the texture, to the touch, and you wouldn't think spores would be able to detach and be glued to it with all that moisture.
Yeah.
But just like 10 minutes or so, we set them on aluminum foil or
Start
wax
stinging
paper.
and
Yeah.
falling off.
Yeah, and it's amazing that can detach from that liquefied
Yeah.
texture on the outside of the
Mhm.
mushroom. Now
I
that's
think
let
it'd be
go.
like tacky or gummy where it would drop off.
That's what they're there for, though, is they want to drop off and be taken somewhere else.
And, is that the same as black mold in people's homes?
So there's fungi are weird. There's so when I say like black mold, basically what I'm talking about is just one particular part of their life cycle, which is after, in this case, when they've made this asexual spore. For Aspergillus, it makes this particular structure that makes that black appearance, but actually the mycelium is white. And it's not until they're ready to put up their spore structures that they do that and make a black spore and do that. It may or may not be similar to the ones that you're seeing at home. There's a huge category of ones that appear this way. One of the ones, you know, a lot of questions I get because I'm a fermentation and bourbon guy as much as I am a fungi guy, is about the whiskey mold. You know, this. Yeah, so that one that, you know, I've gotten called up by CBS, you know, asking for quotes from me on that and stuff. And I'm like, I don't want to touch that. Like, I don't know enough about what this stuff is. And they're always talking about the health impacts. And I'm like, I don't do that. I'm an engineer. And so, but anyway, lots of interest in that. And clearly that's black. But, you know, it's a very interesting metabolism that it has and the ability to metabolize ethanol the way that it does. But yeah, that's a totally different organism than Aspergillus.
What stage of distilling is that happening? Is it that happening coming out of the distillery off gases?
Yes, so it's coming from vapor. So yeah, ethanol vapor. So you'll see it a lot around the rack houses, and also just anywhere where ethanol vapors. It could be, you know, if you have open top fermenters, that's a place where those vapors are going to get taken out, like a vent in a window somewhere, and then get exposed to the sides. And it's like low concentrations of ethanol vapor that I think are the kind of feeding grounds for that particular fungi. So yeah, I guess I've seen it on barrels. I've seen it more commonly on the wood around the barrels than, and I wouldn't be worried about it getting into the barrel. The barrel is at way higher ethanol concentration than
It's crazy. Before I worked at a company, we built equipment for the distillery, and we produced within stuff. Just every tree, everything we had.
that's the story, right? During Prohibition, they could find the distilleries by following the black trees. Yeah.
I thought maybe. Yeah, there's so much, but yeah, even a small.
Do you have any active research being done on whiskey fungus right now?
I don't. I have a sample of whiskey fungus that I took from, it's in a little Falcon tube at home, actually, because I don't want to put it into my biosafety protocol yet. But I have the organism, and I've been wanting to do stuff because that's a killer story, right? Like, use this fungus, we know that it grows well, and then I bet it would do well growing on the stillage substrates that we have too, because stillage itself does have residual ethanol in it. So those stills are not 100%. So I think it would probably do pretty well. It's on my to-do list, I'll say. Yeah, if you want to come into my lab area, you can come and work on it. Yeah, right. I'm always looking for new.
They complain on people for their homes and things, and they always want distillery to be responsible, you know.
Yeah. So
Type of cleaner that, you know.
I think getting it at the source is the best bet, and that's another thing that I've thought about is there's, you know, a lot of helpful things that have come out of like for confined animal facilities, like that form of agriculture. There's biofilters for instance, for dairy barns, for that type of industrial scale of farming. Ventilation systems will often go through a biofilter that can, basically what you're doing is you're getting all the stuff that would grow on the volatile organic compounds and things that are in there to grow in that biofilter rather, and then they clean it up by the time that it gets out on the other end. So I would be, you know, and I have seen interest in the biofilter people in Kentucky that have companies in that are active at the Jim Beam conference that we have every year. So, well, I would call like in general. Technically, yes. Economically, I don't know. Like at that point, it's pretty like the still bottoms. Distilleries do a good job of getting what they can out, but there's some like thermodynamic things with like ethanol, and it makes a special thing in solutions sometimes called azeotrope, and there's like some like issues on getting away from a total recovery. Um, getting away with that is sort of difficult. So economically, I would say probably what you've got going in your distillery is probably about as good as it gets, because distilleries are very incentivized to get as much every drop of ethanol that they can. But still, some comes out in the distillers, some comes out in the vapors. We're talking about like trace amounts usually, and but the category that I was saying, volatile organic compounds, like scientists will just abbreviate that to VOCs. When you're talking about like the smell of the dog odor, those are VOCs. Yeah, so most of the things that we have like visceral reactions to in smells are VOCs, as are a lot of the things that we enjoy the smells of, are VOCs. So in small amounts, they're good, but then in high amounts can be problematic or sensitive to us. Oh, we could talk forever. Wow. It's great. Yeah. So thank you guys for coming. This is really a pleasure to talk with you and have such an engaged audience here. So thank you. Appreciate it. I'll stop the share and stop the. Oh, and I see we have somebody on. I knew it. Yeah. Okay.
Dylan is the vice president of the Bluegrass Mycology Club.
Oh, excellent. Happy to have you here.